this is Roger Skelton again, and this is how hemoglobin works. Points of discussion on this are, first we'll be talking about the structure of red blood cells and hemoglobin, how oxygen binds to the hemoglobin, structural changes that occur in the heme after binding, binding capacity of oxygen in the lungs versus capillaries, how CO2 is bound and transported, and how CO2 interacting with water changes pH. Let's see, red blood cell structure, just real quick. It's the only cell in your body that doesn't have a nucleus. It contains almost no cell organelles. It's disc-shaped and can change shape and bend and squeeze through narrow capillaries. One red blood cell holds 280 million hemoglobin molecules, and then one drop of blood contains about 5 million red blood cells. So it's a lot. Oh, and it's about 70% of the cells in your body are red blood cells, 5 trillion of them. Here's the hemoglobin structure. Each hemoglobin is made of 4 polypeptide chains. That's the globin part, and that's these right here. You've got the 2 alphas and the 2 betas in these chain structures. And then each hemoglobin contains 4 heme groups. There's 1 heme group on each chain, so these are your hemes here. And they're a reddish color because of the iron, and that's actually what makes your blood and muscles red. So let's see here. So each heme contains 1 ferrous iron. That's a +2 iron that's been oxidized already, and that's what oxygen binds to. And then each heme can carry 1 diatomic oxygen molecule, also known as just simply O2, because that's usually how oxygen exists, is in a diatomic form, which means 2 oxygen atoms. And you can see kind of the OChem layout of the heme here, and we'll get more into that in a second. So where oxygen binds to the heme, it actually binds right here on the iron, and you can kind of see, so this is the same thing, but this is kind of flipped this way. See the iron in the middle? So like we already said, each heme contains a central ferrous iron atom. The iron atom is bonded to 4 nitrogen groups. You can see them here. So that means that you have 2 more spots where the iron can make bonds. The bottom spot is called the 5th site, and this is bonded to a histidine, which actually connects to the helix, which we'll see in a second. And then the 6th site is on the top of the heme plane, and that's where the O2 binds. So keep in mind this is like this, and this is where the O2 binds to that top of the iron. And we'll see how you can actually see right here how it changes the form a little bit. As it binds to it, it pulls this up. So it pulls that iron and this up into the plane a little bit more, the heme plane as it's called. So structural changes to the heme, which we kind of just touched on. It lifts the alpha helix towards the heme plane, and this actually changes the interface between the alpha and beta helix dimers. It pulls them up a little bit as well. And this actually makes the other hemes a little bit more conducive to attracting oxygen. So they have a higher affinity for oxygen after 1 or more of the hemes is bound to oxygen. So without oxygen, it's deoxyhemoglobin, and that's in the T state, which is with that ferrous iron a little bit lower, and then with oxygen it's an oxyhemoglobin, and that's in the R state, and that's with the iron lifted a little bit into that heme plane. So, and like we already said, the R state has a higher affinity for oxygen, and we already covered that part. And this is just showing it again, so you can see that it lifts it up into the plane, the T state, R state, and then this shows it pulling on one of the alpha beta helixes. You can see that histidine group down here, and so in the background it's a little bit lower, and then after the O2 binds to it, it's lifted up. And that pulls on this whole helix, which makes it have more affinity for oxygen. On the gas exchange for O2, so pretty much everything in the body, or a lot of stuff in the body, diffuses from either high concentration to low concentration, or high partial pressure to low partial pressure, or based on pH, like we'll see later. So O2 goes from high partial pressure of O2 in the lungs, and that causes it to diffuse into the lower partial pressure of O2 in venous blood, basically, that is passing by the lungs. And then the O2 binds to the heme, and then the reoxygenated blood goes from the lungs to the heart, and then is pumped back out to the body to bring oxygen to the tissues again. And you can see over here, as it comes in, this is venous blood here, or capillary blood, which is mixed venous and arterial technically. But even so, the PO2 is really low, so it's 40. And then the PO2 over here in the lungs, the alveolus of the lungs, which is the little air sacs, the PO2 will diffuse across into that lower one, and then it kind of becomes arterial blood with a PO2 of 100 and a lower PCO2. The PCO2 diffuses across too, but we'll get into that later. Well, right now actually. So this is how CO2 is exchanged. First it moves from the tissues, like your muscle cells or whatever these are here, from a high PCO2 to lower PCO2. O2 moves from the blood, or high PO2, into the tissues, which have a low PO2 at that point. And then CO2 is actually transported in 3 ways. The hemoglobin itself actually only transports about 20%. Our textbook said 14%, but a lot of the internet, the numbers were 20%, 10%, and 70%, so I kind of like those numbers better. So one way that CO2 binds to the hemoglobin is with the terminal amino groups. Remember those nitrogens, and that's the globin part of the hemoglobin. And then once that O2 is off of that heme, the hydrogen ions bind to that heme, which makes red blood cells a little bit acidic, and then makes the blood a little bit acidic. Instead of 7.4, it might be 7.3 or 7.35. So, but that's only 20% of the CO2 that is transported on the hemoglobin. Most of the CO2 is actually transported in the plasma in the form of bicarbonate, or just diffuse CO2 in the plasma, which is about 10%, but 70% is transported as bicarbonate. And it uses this is what we call in respiratory anyways, the hydrolysis equation, which is actually one part of biochem I use at work. So, you can see this bicarbonate bonds with the hydrogen ions, that makes carbonic acid, and then carbonic acid splits into a water molecule and CO2, and then that all gets transported in the plasma, and then we breathe out the carbon dioxide as it gets to our lungs and gets diffused into our lungs. And then this happens across red blood cells too. Red blood cells have a lot of carbonic anhydrase, which does this whole reaction also. So that's pretty much how the CO2 gets diffused through the blood. And then, like we said earlier, pH can also affect everything as well. So it affects the hemoglobin's affinity for oxygen. Active muscle tissue has about a pH of 7.2, which that causes O2 to move from the heme into the muscle, because it goes from that higher pH to a lower pH. Lungs have a pH of 7.4, and then the venous blood going through the lungs has a pH of like 7.3 to 7.35 or 7.37 according to those numbers. So that causes O2 to come from the lungs into the blood. And then another way that the pH changes, like we already said, as O2 is removed from the heme and given to the tissues, it's replaced with a hydrogen ion on the heme, and that makes it that 7.37 or so. And then you can see the numbers over here. Arterial is usually 7.4, somewhere in there, and then venous is, that's actually a little high. A lot of times it's like 7.35 or so, but either way, it kind of demonstrates how it works. And that is all, I think. So thank you.